Welcome back, dear scientist-to-be. I am the Geneticist, and today we're jumping into the coolest, most mind-blowing tool in biology, CRISPR-Cas9. Get ready to talk about editing life itself, with all the nitty-gritty details. And we're back. To truly understand the power of CRISPR-Cas9, we need to meet its incredible team of molecular players with Celi and Cario. This is where it gets fascinating. So, what is this CRISPR-Cas9 magic? At its very heart is the Cas9 protein. Think of it as our super precise RNA-guided DNA endonuclease. It's the ultimate molecular scissors for introducing double-stranded breaks exactly where we want them. The most famous version comes from Streptococcus pyogenes, known as SP-Cas9, but scientists are even tweaking it into variants like SP-Cas9NG to recognize new DNA addresses. Inside Cas9, there are two key nuclease domains, the HNH domain, which snips one DNA strand, and the RUV-C domain, which handles the other. They're both essential for its cutting power. But how does Cas9 know where to cut? That's the job of our superstar guide RNA. In the lab, we use a clever synthetic version called a single-guide RNA, or sgRNA. It's a fantastic fusion of the natural carRNA and trachRNA, forming a single molecule. This sgRNA has a special 20-nucleotide sequence that perfectly matches the DNA we want to edit, plus a scaffold region that helps it cuddle right up to Cas9. And here's a crucial secret handshake. Cas9 can't just cut anywhere. It absolutely needs to recognize a tiny DNA sequence right next to its target site, called the protospacer adjacent motif, or PAM. For SP-Cas9, that's typically NGG. No PAM, no cut. It's like a VIP pass for Cas9 to enter the DNA zone. Oh, and for Cas9's cutting action to work, it needs its trusty cofactors. Magnesium ions, Mg2+. They're like the tiny catalytic engines. And get this, scientists can even use small molecules to control Cas9's activity, or engineer Cas9 to fuse with other enzymes, like a cytidine deaminase for base editing, to expand its awesome genome engineering superpowers. Okay, let's shrink down even closer. How does Cas9 actually perform this molecular surgery? It's genius. When Cas9 finds its PAM doorbell, it gets a green light. The DNA unwinds, allowing the guide RNA to snugly pair up with one of the DNA strands, forming something called an R loop. It's like a little RNA-DNA dance. Next, the guide RNA perfectly base pairs with one of the DNA strands. That's our target strand. This creates a unique RNA-DNA hybrid structure called an R loop, pushing the non-target DNA strand aside like a displaced ribbon. This snug fit makes Cas9 do a little jig, triggering a huge conformational change. Basically, it transforms into its active cutting pose. These changes aren't random. They create a perfectly charged cavity for DNA binding and position those HNH and RUV-C catalytic domains right where they need to be. Then, in a perfectly coordinated ballet, the HNH domain cleaves the DNA strand complementary to the guide RNA, while the RUV-C domain snips the non-complementary strand. Both domains need this precise activation, often thanks to clever allosteric communication within the protein itself. The actual cutting, the catalytic mechanism, is helped by those two Mg2+ ions in the RUV-C active site, breaking the phosphodiester bonds. Specific amino acids, like H983, act as tiny chemical helpers. After the cut, Cas9 actually stays tightly bound to the DNA. This whole precise process, regulated by allosteric control, helps keep off-target effects at bay, but even tiny mismatches can sometimes alter Cas9's dynamics, leading to unintended cuts. It's a delicate dance. So how do scientists actually use this complex system in the lab? It all starts with designing and preparing our guide RNA. We meticulously select our target gene, then either synthesize the sgRNA directly or clone it into special vectors. Next, we prepare the Cas9 components, whether it's the Cas9 protein itself or a plasmid that tells cells to make Cas9. Sometimes we even pre-assemble the Cas9 sgRNA ribonucleoprotein, RNP, complexes before delivery for immediate action. Then comes the big hurdle, delivery into the cells. This is where protocols really get tailored to different cell types, whether they're stuck to a plate, floating in liquid, or even yeast. For induced pluripotent stem cells, hiPSCs, which are super important for disease modeling, A big challenge is their lower efficiency for homology-directed repair, or HDR. We use single-guide RNAs with single-stranded oligonucleotide donors and constantly check their karyotype for chromosomal stability. Careful work, but we can introduce precise mutations. Then there are suspension cell lines, like hematolymphoid cells. They're tricky to transfect. We use transient Cas9 expression and intricate workflows for single-cell cloning to get those homozygous knockouts. When working with primary human adipose stem progenitor cells, ASCs, gene knockout is tough, so we often use lentiviral delivery for CRISPR-Cas9. We confirm those edits with PCR, RT-qPCR, and Western blot, a triple check. Hematopoietic stem progenitor cells are crucial for therapies, and here we aim for high efficiency and HDR. We often go for plasmid virus-free delivery of RNPs, achieving knockout efficiencies up to 75 to 90% and HDR rates over 20%. Tools like RAAV6 and flow cytometry help us enrich for edited cells. For iPSC-derived macrophages, delivery can be inefficient or even toxic to the differentiated cells. But lentiviral transduction gives us near 100% knockout efficiency, making it scalable for huge genome-wide screens. Even tiny numbers of innate lymphoid cells, ILC2s, which don't have established protocols, are being tackled with nucleofection, optimizing every step to maintain cell viability while achieving stable knockouts. Once inside, cells recover, and then we screen and select for our edited cells. This can involve phenotypic assays, looking for fluorescence, or using selection markers. If we need super precise edits, we even isolate single-cell clones. Finally, the crucial validation of edits. We extract genomic DNA and analyze the target site using PCR, sequencing, or special assays like HiBit. And sometimes, we do extra functional validation to confirm that our genetic change actually works as intended. Now, let's talk about making CRISPR even smarter. Early on, a big challenge was off-target effects, cutting in the wrong place. But brilliant scientists have been busy engineering Cas9 nucleases to boost specificity. They've used structure-guided mutations, like in hiPSCs, to reduce its tolerance for mismatches, so fewer off-target cuts happen. And through directed evolution, we now have sniper Cas9 variants that keep their high on-target efficiency, but dramatically lower off-target activity. Some clever engineered Cas9s, like vCas9, even create DNA breaks that favor precise repair pathways, like HDR, over those messy, error-prone ones, further reducing unwanted mutations. It's like guiding the cell's repair crew. And for even more power, scientists are altering PAM recognition. Variants like SP-Cas9NG, SPG, and SPRY can target new sites and show even better discrimination against off-target spots. We even have near-PAMless variants that can target almost any DNA sequence while keeping awesome specificity. But the guide RNA is getting upgrades too. Engineered guide RNAs with added secondary structures can make Cas9 super sensitive to mismatches, boosting specificity dramatically. And get this, simply using truncated guide RNAs, shorter ones, can reduce off-target activity by up to 5,000-fold without sacrificing the on-target cut. Plus, smart computational design helps us pick the best gRNAs. They're even fusing Cas9 to inhibitory domains or anti-CRISPR proteins to kinetically insulate on-target from off-target editing. And for ultimate control, inducible Cas9 systems, like those activated by doxycycline, allow us to give the genome a quick pulse-like exposure to Cas9, slashing off-target effects. And to make sure we're always on point, new advanced detection methods for genome-wide off-target activity are always emerging. Plus, clever computational algorithms like CRISPR help us correct for confounding factors in large-scale CRISPR screens, ensuring our results are super accurate. Okay, so we have our super smart, super accurate CRISPR tool. The next central challenge? Getting it safely and efficiently into cells and tissues, especially for therapies. There are several main strategies, each with its own pros and cons. First, viral delivery systems. Vectors like lentivirus, adenovirus, and adeno-associated virus, AAV, are incredibly efficient, both in the lab and in living organisms. They can even target cells that aren't dividing. But watch out, they come with risks, like inserting themselves into the wrong part of the genome, limited cargo space, and potential immune responses. Then we have the non-viral delivery systems. Physical methods like electroporation, microinjection, and nucleofection are great for lab use, but they can be a bit rough on cells and aren't ideal for whole organisms. Chemical methods use things like lipid nanoparticles and polymeric carriers to encapsulate CRISPR components. They're tunable and avoid viral risks, but getting them efficient and targeted in a body needs a lot of optimization. And speaking of nanoparticles, materials like liposomes, polymers, and inorganic nanoparticles are being developed to target specific cells and improve uptake. It's truly cutting-edge delivery. One awesome approach is ribonucleoprotein RNP complex delivery, where we deliver the pre-assembled Cas9 protein and guide RNA directly. This means the activity is transient, reducing off-target effects, and there's no worrying about DNA integration. We deliver these via electroporation or various nanoparticles, and even stimuli-responsive carriers are in the works. And finally, an emerging star, extracellular vesicle-based delivery. Using exosomes or microvesicles, tiny natural bubbles from cells, to transport CRISPR components. They're super safe, transient, and have low immunogenicity. We hope making them super promising for future clinical treatments. Though we're still figuring out large-scale production. Wow. From the tiny Cas9 protein to complex delivery systems, CRISPR-Cas9 stands as a monumental testament to scientific ingenuity. Its precision and adaptability are absolutely incredible, promising transformative impacts from disease modeling and fundamental research to revolutionary gene therapies. The journey to decode and responsibly edit the genome is just getting started, and you're right here with us, dear scientist-to-be. So, if you're as fascinated by the amazing world of genetics as we are, hit that subscribe button right now. See you in the next episode, inside the amazing world of the cell.